Assalamualaikum and hi, my name is Muhammad Sharifuddin bin Faizal. My matric number is S59921. My topic for today is the development of a novel RSIVD-resistant strain of Red Sea Bream. As for abstract, as we can see in the slide, this is the major journal that I have been used to refer to. This journal is talk about development of novel RSIVD-resistant strain of Red Sea Bream by marker-assisted selection with DNA-based family selection. Okay, the main point in this journal is about the production of G1 that was obtained from its apprentice, which is the RSIVD-resistant male microsatellite that was used to identify the DNA parentage analysis, marker-assisted selection using a quantitative trait locus linked to the RSIVD resistant trait that is PMA4_104, and the survival rate of G2 population in 2016 and 2017 that is 78.8% and 83.5%. This is my table of content. As we can see, there is an introduction, problem, objective, method, result and discussion. First of all, let we go to some definition of certain word first. As for marker-assisted selection, the meaning is the method of selecting desired trait in individual for breeding sim that is based on DNA molecular marker. As for microsatellite, it is the short segment of DNA that is repeated multiple times, and it can be used to in genetic linkage analyze to locate a gene for a given trait. As for quantitative trait locking, it is a chromosomal region that contains a gene which can control the trait. For our introduction, marker-assisted selection (MAS) is a most reliable method in order to get specific trait in breeding process. Okay, MAS is the method that will be used in this topic. After that is microsatellite DNA-based parentage analysis was used to determine the RSIVD survivor and have detected that RSIVD resistant male Red Sea Bream specimen genetically relate to most survivor from the RSIVD outbreak. Quantitative trait loci that link to the RSIVD resistant also has been identified, and locus of PMA4_014 has the highest proportion of phenotype variance, which suggests RSIVD resistance was controlled by a single gene that is PMA4_104. The problem statement for this topic is Red Sea Bream iridovirus disease RSIVD is a major cause of mortality for farm Red Sea Bream. Based on the statistic, the total of 79.5% of Red Sea Bream mortality on commercial farm is due to the RSIVD in 2012. Although there is a vaccine that has been produced and already effective, however, the price become the major problem as it will be troublesome for small farmer to buy it, as the cost and labor involved are prohibitive. This is the objective from this case study. We want to produce RSIVD resistant, where Red Sea Bream that is RSIVD resistant can be produced. Also, we want to have a high survival rate fish, and also we want to lower the cost production as farmer can save the cost from buying the vaccine of RSIVD. Next is the general method. First of all, with development of RSIVD resistant strain, the 55 sample population was obtained from survivor of RSIVD outbreak. In 2012, it includes two groups of G1 half-sibling, one from resistant male and second from susceptible male. The G1 half-sibling progeny undergoes microsatellites DNA-based parentage analysis. The G1 individual heterozygous for both PMA4_014 and PMA-MHC2B were selected to be used as broodstock. The segregation pattern of the resistant allele with a positive theoretically showed the ratio of 1:1. As for the field trial, The G2 population was produced from selected G1 in 2016 and 2017 are used for feed trials. There is no mortality due to RSIVD during this phase. However, after 50 days post-hatching, the fish was transferred to fish farm where RSIVD frequently occur. During experiment, every dead fish was examined with RSIVD based by RSIV specific PCR, which the DNA was extracted from spleen. The fragment of caudal fin from the sample from 200 and 192 fish is collected for 2016 and 2017 and was stored in 99.5% ethanol for DNA analysis. Method of DNA analysis. First of all, we will collect the sample, which the sample was obtained from G2 caudal fin. The second step is DNA extraction, where the genomic DNA was extracted with 5% of shellacs. The third step is PCR. Marker PMA4_014 was used for allele evaluation. Marker PMA4_014 was used for allele evaluation as the marker has shown the most significant link with resistant trait RSIVD. The fourth process is separate PCR product, which fragment analyzer was used. Step 5 determine band size, which it will be compared with 25 BP DNA step ladder. Step 6 calculating allele size by using ProSize software. Okay, as for the result, development of RSIVD resistant strain, 43 samples of G1 were identified as progeny for GO, which is its apprentice resistant male, based on microsatellites. Based on marker of RSIVD resistant trait, 22 individuals were selected for G1 broodstock. 2 specimens have died during broodstock culture, which made it become 20 individuals. This remaining individual has been used as broodstock to produce G2. Survival rates of G2 2016 and G2 2017 after RSIVD outbreak were 78.2% and 83.5%. Survival rate for normal Red Sea Bream for 2016 was only 63.8%, which we can see that survival rate of Red Sea Bream that contain resistant traits is higher. During mass mortality, egg sample was collected and showed the presence of RSIVD. The egg sample also collected at the end of experiment for diagnosis of RSIVD, at which time no RSIV was detected by PCR. Result DNA analysis. Okay, DNA analysis was done to see the present genotype of PMA4_014 in G2. As we know, PMA4_014 is the resistant marker. There is 3 types. There is homozygous for resistant allele, heterozygous for resistant allele, and negative without the resistant allele. As for discussion, the RSIVD resistant strain of G2 was completely produced by using MAS method with DNA-based family selection. This G2 population show a higher survival rate of RSIVD in field trial. To develop the resistant strain, the most important key was finding a male broodstock specimen that has survived an outbreak of RSIVD in the field. This is based on Sawayama and Takagi. PMA4_014 marker can be determined as RSIVD resistant trait marker. Individual with the resistant allele PMA4_014, both homozygous and heterozygous, showed significantly higher survival than those without the resistant allele. The survival rate of homozygous and heterozygous do not differ significantly, suggesting that PMA4_014 is a dominant allele. Conclusion: marker-assisted selection (MAS) that combined with DNA-based family selection is a powerful tool for breeding disease-resistant fish strain for aquaculture. We can do this by monitoring family structure after mass mortality caused by other pathogen and identifying a major QTL affecting disease-resistant traits that can lead to increased survival of other farm fish species as well as Red Sea Bream. Hence, this technique can be implemented in the commercial production process that will have a disease-resistant strain to be constructed in a single generation. Okay, this is my reference. That's all from me. Thank you for listening my presentation and feel free to ask me any question.